Hello, it's a pleasure for me from the 22nd Congress of the EHA to discuss the presentation as a late-breaking abstract of the JULIET study, which is a CTL019 study evaluating the safety and efficacy of this CAR T-cell in relapsed refractory DLBCL. We do know that there is a considerable unmet medical need in this patient and that CAR T-cells have been developed to try to address this need. I won't go into the details of CAR T-cell technology, and you will find in Lymphoma Hub an interview of my friend and colleague Steve Schuster, who will develop in much more detail this aspect of the study. What I would like to go over with you today is the clinical result of the study. This was a global study, JULIET, sponsored by Novartis, which took place in 4 continents, 27 centers, and enrolled many patients. The results which I will present are the result of the first interim analysis with 85 patients evaluated for safety and 51 patients evaluable for efficacy. Why these numbers? First of all, the JULIET design was different from other studies in which the particularity of the JULIET study is that patients were first harvested to have an apheresis and cryopreserved cells before to be enrolled in the study. In US, actually, it was even 2 separate studies. So this let the physicians the time to continue screening, to eventually provide some chemo to these patients that were highly refractory. And then the patient was enrolled, and there was a manufacturing of the cell, which were again cryopreserved before to be delivered on site. So this gave a lifetime interval in between. Unfortunately, some patients progressed during this lifetime. There is a scale-up in the capacity of the manufacturing, which improved over time, with more than 90% of the patients that had a manufacturing product in due time at the end of the study. When we look at the safety in the 85 patients that received the CAR T-cell CTL019, the safety are quite interesting. If we look at the adverse event of specific interest, such as CRS, neurotox, the CRS was 57%, with about 1/4 of the total number of patients that experienced a grade 3/4 CRS. I should also mention that the grading of CRS may differ from study to study. In addition, about 20% of the patients that had neurological event, grade 3 or 4 also. All of the CRS and neurological events were reversible with appropriate management, and there were no deaths related to the studies, to the CTL019 infusion, and no cerebral edema that were observed during the study. So it's quite an interesting safety profile. About 1/4 of the patients had neutropenia, prolonged neutropenia or infection, usual in this category of patient. So a good safety profile. In addition to that, when we look at the efficacy, the overall response rate, best overall response rate, which was the primary endpoint of the study, was 59%. And if we look at the patient, 51 patients that had been observed for at least 3 months, the response rate was 43%, with 37% of the patients being in CR. This is very encouraging because based on the previous experience of University of Pennsylvania, these patients that have reached a CR at 3 months appear not to have relapsed. So it's a little bit early to confirm this result, but this is very encouraging. And as a matter of fact, the estimated duration of response is of 79% for this population. So these are early results, but in a very difficult to treat population. I should recall you that half of these patients in the JULIET study had relapsed after autologous transplant. About 27% of the patients were double hit or triple hit patients. So very difficult to treat population. This is a very encouraging result. Based on this result, we do hope that these CAR T-cell facilities will be available for some of the patients, but also that we'll continue clinical trial, such as randomized trial in the future with the different CAR T-cell being available.